Okay, so in this video we will talk about the internal morphology of the coralloid root. So we had already discussed the internal morphology of the normal taproot in Cycas. So now we will talk about these coralloid roots. Now, the internal structure of the coralloid roots is similar to that of the normal root. The only difference it lies in the poor development of the secondary growth. Yesterday, in my previous video, we had discussed on the secondary growth of the normal root. But in case of coralloid roots, this secondary growth is poorly developed, and also you already know the second difference is that in coralloid roots we know the presence of algal zone in the cortex, okay, which is absent in the normal taproot. Now in case of coralloid roots, here you can see in this diagram here, this is the outermost layer. All right, this is the outermost layer, and the outermost layer here, it is single layered epiblemma. All right, it is single layered epiblemma which may become ruptured due to the formation of this periderm, that is the corky outer layer. All right, the corky outer layer of the root, the periderm. So due to the formation of the periderm, the epiblemma, the outermost layer, it may become ruptured, and also a few lenticels are also developed in the periderm layer. The lenticels, okay, they are present in the periderm layer, and this cortex, as you can see just below the first layer and this layer below here, it is the cortex. And as you can see in the picture, the cortex is white, and it consists of the parenchymatous cells, okay. But not only that, as you can see here, all right, this is the first layer, the epiblemma. Just below the epiblemma we have the cork, all right, which will give rise to, that is the periderm. Then this layer from here till here is the cortex. So it is very wide, but it can be divided into three distinct layers, three distinct zones. Now this outer cortex here, all right, this outer cortex, outer portion here, it is composed of compact polygonal parenchymatous cells, okay, these cells which are polygonal and parenchymatous. And the middle cortex layer here, you can see this is the outer cortex, then we have the middle cortex. All right, this middle cortex is also known as the algal zone. All right, the algal zone or the middle cortex is one cell wide. You can see here one cell wide, and it consists of loosely packed, radially elongated, thin walled cells with large intercellular spaces. All right, these spaces between the cells is very large, and these excuse me, here as you can see clearly in this picture that these spaces, they are occupied or they are inhabited by the colonies of the microorganisms such as the endophytic blue-green algae, cyanobacteria, Anabina, Cycas, nostoc, and besides some other endophytes like Oscillatoria, then diatoms, and some fungi, and also few bacterial species such as Bacillus, Azotobacter, and so many other. So all these, they inhabited these intercellular spaces in the middle cortex. All right, in the middle cortex, also known as the algal zone here. And the last layer here is the inner cortex. All right, this is the inner cortex, and in the inner cortex you can see it consists of thin-walled compact parenchymatous cells, whereas the structure of the endodermis, the pericycle, the vascular bundles, and even the secondary growth is the same, okay, as in case of normal roots that I had already discussed in the previous video. All right, everything is the same. The difference is only that the secondary growth in coralloid roots, it is poorly developed, and even the presence of this algal zone, okay, which differentiates it from the normal tap root. So this is the internal morphology of the coralloid root. So in the next video we will discuss on the internal morphology of the stem.